So today I'm going to talk about molecular insight into μ-Opioid deactivation. And this is the work that we did in Stanford University in Pander Lab at chemistry. So the content of my talk is actually distributed among two topics. The first one is the μ-OR deactivation and ligand-induced fit, which I'm going through some kind of nice things about the activation processes and the activation of the receptor. And another one is one of the challenging questions that we are addressing about the binding pathway of the drug to the receptor. Why, μ-Opioid receptors are important? Because it's one of the oldest actually drug targets in pharmacopoeia, and people actually try to design drugs, which are like analgesics, which can basically sedate. Drugs like Tylenol actually bind to this receptor, and they are just creating a kind of downward cascade inside the cell. As you can see, the morphine, which is a classical type of opioid, is binding toward here, and then it is triggering for the GPCR, actually for the G protein to bind here, and then after that it's gonna just activate the cycloamp. But the problem with most of the analgesics is that it is going to make some kind of bias signal which causes arrestin actually to bind, which it creates some kind of adverse effect. So in drug design, mostly one of the challenges is the type of side effects that we get for a drug. So to solve this problem, the computational powers that we have is really great, to address such kind of question that at molecular level, using these computational microscopic tools that we have, like molecular dynamics, quantum chemistry, how can we find that such kind of very, very tuned type of biologically evolved receptors can become some kind of, sometimes, arrestin bias, or some kind of a specific ligands like morphine bind to this receptor, and some of the other ligands, which are the cardiovascular drugs, are not. So also to mention, this is heavily expressed in the brain. We have a bunch of these receptors right now in your brain, which are trying to just, send signals in order to sedate. So they are heavily expressed in CNS system. So as I can say, as Uncle Ben actually told Spider-Man, with great power comes great responsibility. For us, with great blue waters, we have great computational powers, which comes great problems to challenge, and challenges to solve, like drug design or computational, basically exploration of the proteins. So to just give you a kind of idea what this receptor looks like, the structure of μ-Opioid, fortunately, at Stanford University, we have Brian Kubilka, who won a Nobel Prize 2 years ago, on the crystallization of this kind of receptors. The active structure, which is pretty difficult to crystallize, it's very fluffy because the G protein is bounded there. And there is a very, very fine type of conformational changes inside this protein when the ligand is binding. So the co-crystallized ligand, which is bound here, is called BU72, which is a high affinity, actually, type of opiate. So, they put the ligands here, and the G protein actually bound downward in the cytosol. And then there is this 10 angstrom movement of the TM6, which is observed. But there's no detail of the intermediate steps which this ligand actually creates in order to activate this receptor. So what we can do is that using computational powers that we have, and also we have developed a kind of code called TCOD, Transient Independent Component Analysis, which is a machine learning code, which is like a detective. So what does this detective do is that basically it is like a kind of magnifier, going over all the residues and amino acids that we have inside the protein, and it looks at the which of them does a suspicious type of conformational changes. For example, if the tyrosine, one of the amino acids, is just flipping or something like that happens, this TCA, which we have it here, and also it's on the GitHub, you can just fork it and use it. It's basically orthogonal to the PCA, principal component analysis. And then for this part of problem, we used the actually main receptor in the active state. We took the ligand out, and we run something around 178 microseconds of all atom MD simulations. And then what we get is a kind of, we found a very interesting intermediates, actually. So this is the free energy landscape we explored through all this data using TICA and MSM, Markov State Model, which we built out of this receptor. This is in active state right now. And look at this tyrosine, which is just in the active state. So it explores all the possibilities in the active stage, as you can see. And then at one point, I mean, it just discovers a kind of well inside the intermediate step, which you can see this tyrosine from the crystal structure. We observe that it is just flipping. And this flip is pretty important for the deactivation process, and it's not available in the crystal structure. And then from here, actually, the receptor is traveling toward the full inactive stage, as you can see. I mean, it's exploring all different possibilities. And the marker to see that the receptor is fully deactivated is that TM6 is moving fully toward TM3, and you see that distance is decreasing, so by the clusters that we build on. So we got a complete picture of all the intermediates and what is happening through, I mean, clustering and using machine learning in order to find what are the other clusters. In another effort, we run the simulations with APO, I mean, ligand from the active crystal structure out, and we put also the highly active co-crystallized ligand BU72, and we have this, thanks to blue water, I mean, we have this 480 microsecond. I mean, those people who are running MD know actually this type of sampling is pretty difficult. And we got very interesting insight, I mean, toward the canonical three-state behavior, in which when the drug is inside the receptor, we found that there is a kind of another state which is modulated before going to the fully active states. So as you can see, I mean, TICA is a machine learning algorithm that discovers on the fly what are the suspicious type of actually conformational changes, or what are the important reaction coordinates with it is moving. So we found this, I mean, active crystal structure here in the free energy landscape, and there is a kind of new state here. You can see, I mean, there is a kind of well created, and that new state is actually mediating type of moving toward the active crystal structure, which is a kind of induced fit between the ligand and also tyrosine 326. And as you can see, I mean, when the ligand is just present there, I mean, there's a kind of lock-in type of behavior in the ligand, which it goes and sits here. I mean, it adjusts itself to the neighboring residues, which is a tryptophan 293. And there is an interesting phenomenon with the tyrosine 326, which is moving and aligning itself toward the drug. And another important fact that we found in the induced fit is that when the ligand is present, there is this third state, which is very interesting, which is this tyrosine down from the look. If you look, I mean, this is the active structure in orange, and this is the fully inactive structure, the structure with the ligand. And you can see, I mean, this tyrosine is going down, is a kind of switch for the activation of the ligand, for the activation of the receptor. So another problem that we tackled with actually using blue waters is a kind of twofold question. The question is that when you have the drug inside the blood, or it's in the solvent, how it is just attracted toward the receptor. And another important thing in drug design is that why, I mean, a kind of cardiovascular drug is not sitting here in the opioid receptor, why this specific chemical compound is just attracted to this. So we did around some kind of, I mean, thousands of simulations, and we found out that there is a kind of selectivity region here, which is very important. I mean, there is a kind of amino acid sequence from 310 to 315, which is like, I mean, a radar on the extracellular part of the receptor, which is just, we call it fish hooking mechanism. It just selectively hooks to opiates, and it is selecting the opiates. So if you have a kind of other compound or drug, it is not going to be bound to that. So I have a kind of movie here that you can see that how this guy is embracing the ligand here, which is a kind of opiate, is a high. So again, and this is the type of interactions that it creates. So it's like a kind of lock on the top part of the receptor, which is responsible for selecting the type of ligand. And then from there is another question that how basically the ligand is moving toward the orthosteric site. So we call this site allosteric, which is the kind of extracellular type of site. And then the ligand, we didn't see that the ligand goes through a slingshot inside the orthosteric site, which is somewhere down here. So it gets some kind of, orientational effects, and then after some time, it gets into the receptor. And also we did some parameterized ligand, using the force field toolkit, using quantum chemistry. Gaussian software, it's very painful, to just parameterize these ligands, using Gaussian, thanks to force field toolkit in VMD, which is a great tool, I guess, in order to do this. So also to say that this is a selective actually filter here, we did a kind of competitive binding analysis between all different type of drugs. And you can see that IBNTXA, which is the kind of drug which for the 6th helix splice variant of this receptor discovered in Memorial Sloan Kettering, is not binding to this receptor with the 7th transmembrane helix, which is pretty interesting. And we found that the time to bind, which is the time that from the solvent we have this ligand coming and just selectively attached to the allosteric site, is around 25 to 30 nanoseconds, and there's a distribution here. And also in another study, which I don't have the plot here, we put some kind of cardiovascular drugs here in the solution, and it didn't actually bind to this selective allosteric site. So, but another question which is remained unsolved is that, okay, we know the allosteric site inside the μ-Opioid receptor, we have the orthosteric site. What is the path and what is the time scale that it takes to go there? So we run these simulations for 3, 4, 5 microseconds, and we didn't see this going through and just bind into the orthosteric site. So we thought that we should just generate a lot of simulations from here with different orientations along the path between the two A and B positions, which are basically allosteric site and orthosteric site. So we generated different seeds here, and as you can see, these seeds were simulated, each of them around 1 microsecond to 2 microseconds, and we have got the aggregate of 150 microseconds here. And we found a very interesting network. This is created by MSM, Markov State Models. And we found, there are two stable allosteric sites, which I talked about one of them previously. And the ligand takes a lot of different pathways, to go to the orthosteric site, which is down here, which creates the activation of the μ-Opioid. And the interesting part, this is a kind of MSM movie. It shows that how ligand goes through the very, very tortuous path that it has from the allosteric site to the orthosteric site. So thanks to the computational power that we have, we could just, the path that, the drug takes in order to go to the orthosteric site. So, and also we found some kind of very, very high probability passes using Markov State Model. As you can see, there are 2 allosteric sites. There are 2 potential wells, and there is very, very well-defined orthosteric site, which is a very deep well because it's a place that the ligand should be. And there are kind of pathways. Using TPT, transition path theory, we just found out this path. So in conclusions, we found out that intermediates of the receptor, the activation and pathway is understood. This is very important in the drug design and the way that you can just find and try to find the hits and the hits to lead. because when we know the activation of the mechanism of the activation, it helps us in order to design actually the type of drugs which are great analgesics, and they don't have the side effects. By side effect means, they are not arrestin biased. The allosteric site of MOR and its drug hooking mechanism is found, so we didn't know where is this selectivity filter that filters out different types of drugs. And the allosteric site is selective to the opiates, which we showed. We put different kind of opiates in a mixture with the different type of other drugs, which are not supposed to bind, and we found that selectivity is not coming from the orthosteric site, but it is coming from a kind of upstream allosteric sites in the extracellular part. And this induced fit originated from the co-crystallized ligand is stable in the third state, which is the tyrosine down, which is the new state which you cannot see it in the crystal structures, neither in the inactive crystal structure nor in the fully active crystal structure, and it's a kind of intermediate along the path. And that is quite necessary for the activation mechanism. And we are collaborating with the Stanford Neuroscience and the Stanford School of Med in order to work on some kind of beta 1 and beta 2 type of receptors, which are the heavily cardiovascular drugs. We are trying to find some kind of we developed a kind of machine learning algorithm in which it can just find out what type of drugs are basically arrestin bias and creating some kind of side effects, and what kind of drugs are fully GPCR or G protein actually activated. So acknowledgement, we take Halloween pretty serious in Stanford, especially at Vijay's lab. So this is the Halloween last year, our costumes on the Clark Center. And thanks to the Pandel lab, there are great people working here, and Vijay, and also my colleague in the School of Medicine. He's an MD PhD, Ivan, which does a great and fantastic job. And also thanks to the MSM. And I really appreciate the allocations that we have through PIRAX with Blue Waters. Without Blue Waters, of course, most of these were not possible. And also the other thing to mention is that Blue Waters is such kind of nice machine that we have folding at home, we have a lot of clusters at Stanford, but this is really, you can take a lot of nodes, and with that computational power, you can solve challenges in health and medicine. Thanks very much.
Thanks. Hi. So in your graph, like when you have the pathway and
right.
So you show a free energy.
Right.
So how accurate are the maximum in that free energy? So because I can guess in several microseconds you get the minimum, the relative stability of the minimum is probably relatively accurate. But how about the maximum? How often you visit different routes to say, well, this is the real maximum we see, and we can say the path?
That's a great question. So this is a weighted free energy plot. it is taken from the MSM, and actually by the TICA that we get all this sampling. So it's not, real free energy that you get out of, weighted histogram methods or things like that. So it is taken out of the population of the states. So the more population you have, and the thing that here actually responds to your concern is that we normalize it to the maximum actually likelihood. So there's no problem. You can see that around the boundaries, around the edges, those are very, very less populated state. So using the population and the amount of net populations that we have, we just normalize that, and so we are sure that these wells are really well pronounced. And in order to say that we have the convergence on the statistics, also we have a kind of measure called implied timescales, which we see that if there's a kind of, it's like detective for the convergence plus how much statistics we gathered. and if that the statistics is converging on this plot. So MSM takes care of that, Markov State Models, basically. Sure. Yeah.
Yeah, so following up with this question, you showed that you had like 512 simulations for 250 microseconds. Is that aggregate or each?
That's aggregate, yeah, that's
Okay.
aggregate. So each trajectory is normally, I guess, 400 to 500 nanosecond, yeah.
Okay, and then like, as you were mentioning in MSM, like you got the ability to see where you have the lowest accuracy and start simulations that just cover that span. Is that also what you're doing?
Yeah,
in those
we are
regions?
receding, exactly, we call it recede.
Okay.
So we recede around the points that we see the reaction coordinate is moving, I mean, slowly in that direction, so we recede around that. I mean, so half of this simulation, 120 microsecond is, I mean, straight simulations, and the other 120 is recede from different points that we got on the cluster. So we build a kind of initial cluster and MSM on the 120 microsecond data normally, and then we find, I mean, different kind of, I mean, clusters there, and we seed around those clusters later with the additional 120 microseconds. Very good question, yeah.
So maybe just to add to the previous question, so you, if I understand correctly, you have a bunch of simulations, and then you can kind of update where you start them based on, so it explores for some time, and then you reseed from that.
Yeah.
Do you like reapply TICA and then look at the new reaction coordinates? Like does that
Exactly, yeah. I mean, so after reseeding, I mean, we are again running TICA on the whole 240 microsecond simulation, not, I mean, from the place that we started receding. That's a very good question. And another thing which is important is TICA is giving us some hints. And another thing which helps us here a lot is that we have the crystal active structure and inactive structure. So we already have the two very well potential wells, I mean, defined. So we do a lot of simulations around them, and then these two converge. The hard problem with some of the receptors like beta 1 is that sometimes you don't have the active structure, which makes it very difficult. So you should just go from the inactive structure, put the ligand, dock the ligand in the inactive structure, and let the simulation go. And you should also be able to discover the intermediate states and also the full active structure, which there, I mean, some kind of methods like a string theory, I mean, which you take the gradients of the energy with respect to the reaction coordinate will help a lot. Eric?
So as you know, I'm a fanatic about combining physics-based approaches with informatics-based approaches.
Yeah, I know.
And you're defining very, very precisely and I think usefully how potential drugs interact with a particular ligand.
Yeah.
Could you foresee using informatics and structure techniques, sequence and structure informatics techniques, to foresee what off-target effects this drug might have?
Yeah.
Is there other stuff around in the human proteome with which it might interact, and could you predict that, or at least project the possibility of that, before you get into your really expensive clinical trials?
Yeah, that's a very good question. So as I mentioned in the progress part, I mean, what we are doing is that we have all these intermediates, beautiful intermediates, and each of them is acting like a new crystal structure for us. And what we are doing right now is we have a kind of assay data for different kind of compounds, which score very well, for example, and we know that which of them is arrestin biased, or which of them is actually fitting to a μ-Opioid receptor, or which of them is a kind of cardiovascular drugs, and they have a score and ranking. And so we use machine learning in order to match the bioinformatics data that we have from all these structures that we get from the intermediates, active crystal structure, inactive crystal structure, and the ranking of them in the being arrestin bias and non-arrestin bias. And we successfully were able, I and my colleague Ivan, were successfully could just mark down some kind of receptor, a specific conformation that are arrestin biased, which is great. So it means that they have a huge side effects, those compounds. So you can filter out that compounds out. And I should mention that, I mean, you talked about, I mean, using chemoinformatics, bioinformatics, and structural biology. Yes, that's possible via docking and machine learning, I guess, combined together. Like, I mean, you use Glide, a Schrödinger package, in order to dock multiple actually type of compounds, and you get a score and ranking, and you can just work with that ranking to see which drug is much more efficient.
Okay. You mentioned about the next goal of your team at the end of your presentation, right? So how does it increase the complexity of your research that Blue Water can help you better? Yeah,
so Blue Waters is helping us a lot in this regards. I mean, as I mentioned, for beta 1, which is a great, kind of receptor, beta 1 and beta 2 receptors are heavily expressed in heart. And in the School of Medicine in Stanford, people are just striving to make selective ligands only to beta 2 or only to beta 1. But unfortunately, these two crystal structures are 78% homologous. So it's a very fine-tuning type of drug discovery problem to make it. So, since crystal structure is only giving us two states, Blue Waters, using these massive simulations, we can find all these intermediates which are very important in finding the selectivity toward a specific type of compound, which, right now, most of the cardiovascular drugs, unfortunately, they bring sometimes cardiac arrest, while, they're bringing down the blood pressure, which is a great thing, but unfortunately, they bind to the other structures. And we don't know that. Why? This should be, selective to beta 1 or beta 2. Why is it selective to, why is not selective to one of them? And how can we tune the type of compound? How can we just seize a specific compound? Because if you put one CH3 group, it becomes a poisonous ligand and it will kill you. So it's very, very fine tuning actually thing for the drug discovery. And Blue Waters can help, in order to get all these statistics and find these beautiful new intermediates which are modulated through these ligands. And we can find the selectivity, the mechanism of selectivity.